The visual system transduces two features of the light wave itself, the wavelength and intensity. The wavelength is the distance between successive light waves, which our visual system interprets as color. The distance from wave peak to wave peak is measured in units called nanometers, nm, which are 1/1,000,000th of a meter. If light waves are very close together, in the 400 nanometer range, we perceive the color violet. And if the waves are far apart, in the 750 nanometer range, we perceive red. And of course, wavelengths in between these two extremes allow us to perceive the range of other colors within the visible spectrum, as you can see here. Humans are able to visually detect only light waves within the visible spectrum, but we can feel the effects of ultraviolet rays, which have a wavelength of less than 400 nanometers, on our skin as sunburn. Rattlesnakes can see light in the infrared range, which is greater than 700 nanometers, helping them to identify prey. The intensity of the light waves is transduced by the visual system as brightness information. The higher the intensity of the waves, the brighter the light is perceived to be. Of course, we perceive much more than color and brightness. Complex features of our visual world are processed by the brain, not the eye. First passes through Laura's cornea, the clear outer covering of the eye. It then passes through the pupil to the lens, which bends or refracts the light to focus the visual image on the retina. The lens left-right reverses and inverts the image. The retina, located at the very back of the eye, is a three-layered structure, and the first place in the visual system where the light energy is transduced into a neural code. Processing begins at the deepest layer of the retina, which we'll call layer 1, and proceeds forward to the second and third layers of the retina. The furthest layer of the retina contains cells known as photoreceptors. There are two kinds of photoreceptors, rods and cones, which are each named for their shape. The rods are located primarily in the periphery of the retina, and they operate best under dimly lit conditions, which means they help us to see in the dark. They provide somewhat blurry images, however, and little to no color information. This is why it's hard to differentiate between colors in the dark. Our peripheral vision also involves input from rods. Cones are located primarily in the center of the retina, on the fovea, and they operate best under well-lit conditions. Cones help us to see objects clearly and allow us to see color, which is why it's easy to see colors during the daytime. To remember what cones do and their location, just think of the letter C, which stands for cones, clarity, color, and center of retina, which translates to the center of our visual field. We're able to see everything around us, from our cars to our friends to the messages on our Facebook page, starts with one major thing: light. The objects around us either absorb or reflect light to varying degrees. The reflected light waves of electromagnetic energy stimulate receptors in our visual system, allowing us to perceive those objects. Here's how it works. Let's say 12-year-old Laura is playing soccer and she is looking at the ball. Light striking the soccer ball is reflected back to Laura's eye in waves, which are translated, or what is called transduced, into a neural code that our brains can understand. Since it's a bright, sunny day on the soccer field, Laura's cones are quite active. As Laura is keeping her eye on the ball, the image of the soccer ball is focused on the fovea of Laura's retina, that is, where the cones are located. The soccer ball appears very clear. The fans in the stands, however, are in Laura's peripheral vision and appear slightly blurred because their images are focused on the rods, located in the periphery of the retina. Once processed by the rods and cones, information about the soccer scene is sent forward to the bipolar cell layer of the retina. Bipolar cells use the neurotransmitter glutamate to communicate. There are two types of bipolar cells, off cells and on cells. Off cells depolarize to light off or dark objects on light backgrounds, For example, the black pattern on the white background of the soccer ball. On cells depolarize to light on, or light objects on dark backgrounds. For example, the white portions of the soccer ball on the much darker green grass. Each bipolar cell responds to a tiny portion of Laura's overall visual field in a center versus surround fashion, meaning that each bipolar cell's response in the center is opposite of that in the surround. The bipolar cell response to the visual field begins when an individual cone sends information about a small portion of the visual scene it is responding to, in this case, the edge of the soccer ball, onto the bipolar cell layer. The bipolar cells receive the information about the edge of the soccer ball from the cone and compare it to information received from adjacent cones. The bipolar cells then enhance the differences between information received from multiple cones. So, if one cone is responding to the soccer ball and adjacent cones are responding to the green grass, the ball information is in the center of the visual field, and the grass information is in the surround. The bipolar cells enhance the difference between ball information versus grass information, which enhances Laura's perception of the ball distinct from the grass. The third and most forward layer of retinal cells is called the ganglion cell layer. Much like bipolar cells, ganglion cells respond to information in their receptive fields, the portion of the visual world to which an individual photoreceptor responds, in a center versus surround fashion. This means that the response to the center is the opposite to that in the surround. The center versus surround organization enhances our perception of borders and contrasts and helps us to pick out objects from backgrounds. As a result, Laura is able to see the edges of the soccer field and pick out the ball from the surrounding grass. After leaving the retina, information travels to various brain regions, including the superior colliculi in the brainstem and to the thalamus. On its way to the thalamus, the optic nerve crisscrosses to form a large white X, known as the optic chiasm. Chi means X. The shiny white appearance of the optic chiasm is due to its heavy covering of myelin, which helps to speed up the neural conduction or information transfer. As a result of the crisscrossing of the optic nerve, all information from Laura's right side of the world, or right visual field, will be processed by the left side of her brain. All of the visual information from Laura's left visual field will be processed by the right side of her brain. Axons from the nasal halves of each retina crisscross to the opposite or contralateral, lateral geniculate nucleus, or LGN region of the thalamus. Axons from the outer sides of the retina send information straight back to the ipsilateral or same side lateral geniculate nucleus region of the thalamus. Nucleus, or LGN, is the area of the thalamus that processes visual information. The LGN has 6 layers, with alternating input from the left and right eyes. Input from each eye is kept separate. Layers 1, 4, and 6 of the LGN receive input from the contralateral eye. Layers 2, 3, and 5 receive input from the ipsilateral eye. Layers 1 and 2 contain magnocellular, or M cells, which are sensitive to movement, but not color or detail. Layers 3 to 6 contain parvocellular, or P cells, which are sensitive to color and detail, but not movement. A third set of neurons, located ventrally to the M cell and P cell layers, known as coniocellular neurons, are involved in processing color information. Visual information is sent to the primary visual cortex. The primary visual cortex, also known as striate cortex or area V1, is located in the occipital lobe in the very back of the brain. This region contains a retinotopic map of the visual field. Neurons in the primary visual cortex respond to very specific features in the visual field: texture, line orientation, line movement, periodic patterns, and spatial gradients, to name a few. The rich details and textures of the visual scene are analyzed here, but the big picture or whole visual scene does not yet come together for Laura because information is not combined here. The role of the primary visual cortex is feature detection in small segments of the visual field. For example, edges of stimuli, contrast between the black and white portions of the soccer ball, contrast between the color of the uniforms of Laura's opponents versus her teammates, and the direction of movement of the ball are all features that are analyzed by the primary visual cortex. The cells in the primary visual cortex are arranged in horizontal layers and perpendicular columns. As in the thalamus, information from M cells and P cells are sent to separate layers of the primary visual cortex. Perpendicular modules consist of cells organized in functional units known as ocular dominance columns and blobs, which are involved in color processing. Of the primary visual cortex analyzes the features in one tiny part of the visual field. For us to perceive objects and entire visual scenes, the information from these individual modules must be combined. That combination takes place in the visual association cortex. The visual association cortex, also known as the extrastriate cortex in the occipital lobe, is directly adjacent to the primary visual cortex. Here, Laura begins the process of identification and recognition of visual objects. Two additional components of the visual association cortex include the ventral stream, which is located in the temporal lobe, and the dorsal stream, which is located in the parietal lobe. The ventral stream receives shape, color, and movement information from M cell and P cell pathways. It is here that object recognition is processed. Laura uses the information from her ventral stream to recognize that the soccer ball is being passed to her and that the goal is wide open. The dorsal stream's input comes primarily from M cell pathways. This stream processes movement information and integrates movement and vision. Thanks to a functioning dorsal stream, Laura is able to make contact with the ball with her foot, run toward the goalie, and score a goal.